Okay, we're going to have a bit of a look here at polymerase chain reaction, or PCR as we usually call it. And a PCR machine is a little machine like this. Basically it's designed for thermal cycling, so it goes through a different range of temperatures for the purpose of DNA amplification, so to make one or a few pieces of DNA into lots of pieces of DNA. Imagine if we're working doing some particular tests, maybe doing some forensic tests. If we can extract one piece of DNA, that might be enough if we can replicate it enough times to do all the tests that we need to do. And likewise, if we need to use DNA in some particular experiments, if we had to just do one piece of DNA at a time, it would be a very ineffective way to work with DNA. So having thousands and thousands of copies. And scientists can actually make, in a couple of hours, can make millions of copies of a particular segment of DNA. And usually what they do is they cut what they need with restriction enzymes first, pop it in the PCR machine, and away we go. So to start with, we might have a look at what we might put in our little test tubes here. And what we're going to put in is, well, we're building DNA, so we're going to have our original strand, which we want to replicate. We're going to need lots of nucleotides to build it. We're going to need lots of primers to get things started. And we're going to need the enzyme to add these nucleotides to the new strands. And we use an enzyme called Taq polymerase, and that is the polymerase enzyme that comes from some bacteria that live in thermal springs. It's particularly useful because it can withstand high temperatures, and we'll have a look at why that's important in just a sec. So to just review our purpose here, it's really to go from one template strand and to go through what we call amplification, where we end up with lots and lots and lots of strands, or millions and billions of strands. And it's really a case of every time we go through a cycle, we double the amount of DNA. So very, very quickly, we can end up with lots of copies of the same strand. So let's use this graph here to have a look at what basically happens during this process. And as we mentioned earlier, it's basically thermal cycling. And what we do first is we take the temperature up to about 95 degrees, and at that temperature we're able to split the two strands of DNA. The hydrogen bonds in the middle here that are holding the strands together break apart, and we call that denaturing. And it's not the same as denaturing in proteins, where the molecules change permanently and the shapes change and things like that. Denaturing in DNA means the strands split apart. So that happens at about 95 degrees. Then we drop the temperature right down to 55 degrees, and that's a good temperature for the primers to attach or to anneal. And once those primers have attached, we then bring the temperature up to about 75 degrees, and that's optimum temperature for the Taq polymerase to work. And that's where we'll have the primers being extended with nucleotides to make two complete copies of the DNA. That's one cycle. And then we go into the next cycle, where we go up to 95 degrees, back down to 55, back up to, I think it's actually 72 degrees. And we continue through that cycle. Each time we're splitting apart the DNA, adding primers, extending the primers, extending the strands to make a complete copy. So back to our original diagram, you can imagine that every time we go through one of these cycles, a little up, down, up again, we have a complete reproduction of our strands. And we go through the cycle, we go up 95, down to 55, up to 72, and then we do it again. Every time that happens, that thermal cycle happens, we amplify our DNA, we reproduce it once. Okay, good luck with studying PCR.